Good morning, ladies and gentlemen. My name is Phil Gattone. I'm President and CEO of the Epilepsy Foundation, and I would like to welcome you to our third biannual Epilepsy Pipeline Conference, this time, for the first time, hosted by a newly merged Epilepsy Foundation and Epilepsy Therapy project. And I'm just very, very thrilled to be with you and to welcome you all here today as we begin what will be a thoroughly enjoyable time together. I would like to recognize some individuals and corporations who help make this special time together possible. I would like to first thank Synovian for their leadership level sponsorship as our new therapy innovator sponsor. Synovian has made a huge impact in our ability to serve and meet our mission. So I want to thank them for their leadership and their support today. I'd also like to thank ASI and UCB, who sponsored this conference at the new therapy development level. They have also been key partners of ours for many years, and we are thankful for their support as well. And we have a series of supporters who have really generously given their resources to help this conference remain a success and grow. And those sponsors are Upshur Smith, Visual A's, Cyberonics, the Danny Did Foundation, QuestCore Pharmaceuticals, Medtronic, Lundbeck, and Open Concept Systems. Thank you all very much for your support. One note, Open Concept Systems is the support for our app. So if you have not downloaded the app, you can do so by going to your app store and just typing in a search, Epilepsy Pipeline. And you will have an app that will provide all of the information you'll need and will be very important when we have our vote tomorrow for the Shark Tank. So please. Yes, Epilepsy Space Pipeline, and you will see it, and I think you'll really enjoy that app. I also would like to recognize some of the leaders we have in our audience. We have affiliate Epilepsy Foundation
individuals living with uncontrolled seizures. And beyond SUDEP, seizures and epilepsy take far too many lives. I have the privilege of saying welcome and thank you to all of you who are gathered with us for this exciting two-day showcase of new drugs, devices, and therapeutics, explorations of trends in research and therapy development, of regulatory paths, of financing new therapies, and partnering to bring new therapies to those who need them. I speak both on behalf of the National Epilepsy Foundation and our 47 local affiliates and chapters, and as the father of a 17-year-old girl, Sylvie, who lives with epilepsy, still challenged, but now doing better thanks to new emerging therapies. So I want to say thank you to this wonderful community of researchers, academics, NIH and industry participants, investors, and advocates, so many of you who I have come to know and to appreciate and to count as friends over the last dozen years. I want to thank all of you who are with us today and in this battle, but in particular our faculty and our scientific leaders, Jackie French and Roger Porter, for the rich program we have in front of us. I also want to give my thanks to Marianne Brody and the Epilepsy Study Consortium team, who are our partners in this conference, and to Karen Bergman and Susan Pietrapello for their help and work, and also to Phil Gattone and the Epilepsy Foundation for their contributions and efforts in support of the conference. But I want to extend a very special thanks and appreciation to someone who appears to have just left the room, Kim Macher, Vice President and Director of the Epilepsy Therapy Project of the Epilepsy Foundation, for her role as the chief organizer of this conference in these 2 days, for her incredible dedication and caring and work. Thank you. Let's have some fun and enjoy this tremendous array of ideas, people, and future therapies we have in front of us over the next 3 days. Thank you, Jackie.
Thank you. It's just wonderful to see the room so full and everybody so interested. And as always with these conferences, we have a mixture, and I can look out into the room and I see a mixture of people from academia, people from government, people from former government, people from industry, and of course, people with epilepsy. So this is the audience that should be here, and this is the audience that is here, and we have a full room and we were sold out. And if there's, anything more that can be said, that people want to see new therapies and they want to see these move forward. And I want also to tell all of you that the epilepsy community and the collaboration between all of these groups is now being looked at by other disease areas, saying this is what we should model ourselves against. These people are doing it right. How can we do it the way they're doing it? And I think that is a fabulous thing to hear from other disease organizations. And as far as this conference is concerned, you would like to think that this was like very regimented and organized and everything was done a year in advance, et cetera, but that is not in fact the case. This conference and what you're going to see in front of you grew very organically from the partnership between everybody that Warren was talking about. And, it was sort of like growing organically up until the very last minute. We're kind of laid back people, and we say, oh, here's another interesting thing for us to show. Let's find a little space in the conference and put it in. So that's why you saw the program mutate over time, because we wanted to bring you the latest and the greatest up until the very last minute. And I think we've done that. And I think that over the next 3 days, you'll see that. And the last thing that I want to say is that just as somebody who's got their ear to the ground in terms of new therapies in epilepsy, that over the last 6 months, I have seen more activity in this space than I probably saw in the year and a half before that. For some reason, whatever we're doing, it's working and people are coming back. You know, maybe even a year ago when we were at this conference, there was a lot of people are moving out of epilepsy. They're moving away from epilepsy. How can we get them back in? And you'll hear that a little bit probably over the next few days. But I'm here to tell you that they are coming back. There is an interest. And we're going to hear a panel that's going to tell us maybe a little bit from the industry, private sector, why that might be the case. But it is an exciting thing. Just like in this room, we have these pillars that are obstructing some people's view. We do have roadblocks ahead of us, but, but because of all of the people that are in this room right now, we're going to overcome those roadblocks and get new therapies to epilepsy. And I am the first speaker, so I'm going to introduce myself. Here I am. And my job in 15 minutes or less, which is really not a whole lot of time, is to give you a little bit of insight into the annual state of the pipeline as I see it right now. So I don't know if this works. I'm pressing the forward advance button, but nothing is forwarding and advancing. Ah. Okay. Is it going to work now, or do you have to stand there? It's okay. No, I mean, you're welcome to stand there. So, where are we going and what do we need? So, what do we have now? You hear this mantra that we have treatment for 2/3 of patients with epilepsy. That is, we can get their seizures controlled 2/3 of the time. But I will remind you that those therapies have to be taken daily, which is an incredible burden for people with epilepsy. Even if they have rare seizures without medication, they still have to take their medication every single day. And some people, even if they miss a single dose, are at risk for a seizure, which is really an enormous burden to carry around. And the other thing we have to remember is up until this point, and I think it's related to the last point, our therapies are all symptomatic, which means we are giving a medicine to suppress seizures, but we have no medications that actually impact on the underlying disease. So therefore, what do we need? We need treatment, certainly, for the therapy resistant, and that is where most of the thought and effort goes towards treatment for the therapy resistant. But we do have many other needs as well. We need treatment for difficult syndromes such as Dravet, Lennox-Gastaut, infantile spasms. These are syndromes where we're not talking about 2/3, 1/3. We're talking about basically 100% treatment resistance or 95% treatment resistance. So certainly we need treatment for those very difficult patients. We would love to have an ability to predict what patients are going to respond to which drugs so that we have personalized therapies. And somebody put it very well, from blockbusters to mini-busters. You know, a drug that would be incredibly useful for a small subset of patients as opposed to a drug that you can give to everyone but doesn't work quite as well. We need improved options for newly diagnosed patients. So I don't want those people to be forgotten about. You know, sometimes when I give a talk, I say, I represent the 66%. And we can't forget about the 66% when we're talking about the 33% that are treatment resistant, because they do not represent a satisfied audience. You know, even though they may not have any seizures, they have that at the expense of sometimes significant impact on quality of life, whether it be changes in mood, irritability, sleepiness, difficulty concentrating, difficulty falling asleep, many, many other things that I hear from my patients. So we definitely need new treatments that don't impact on quality of life. And we need attention to comorbidities. We need drugs that actually address the comorbidities such as depression and cognitive slowing and memory impairment rather than making them worse. And as I mentioned, we have a great need for drugs that actually impact on the epilepsy, either anti-epileptogenic or disease modifying rather than just treating the symptom. So what's going on right now? I found this very interesting pharma report of drugs in the pipeline from 2013 for CNS disease. And here we are, the little bar. This is epilepsy. And obviously, this does not correspond with the incidence and prevalence of the disease in our world. But even more so, take a look at the size of those bars in terms of phase 1, phase 2, and phase 3. And which bar do you think is the skinniest? Phase 2. Phase 2 is the skinniest bar. And that is a problem for me. So we have some drugs that are nearing the clinic, but what's going to be behind them to keep our enthusiasm up and bring new therapies to people? So these next few days, hopefully, we're going to find some drugs that will increase the size of that blue bar. I searched on clinicaltrials.gov because I'm supposed to be talking about the state of the pipeline for all of the studies that are in phase 2 and phase 3 for new chemical entities in epilepsy. And I may have missed one. If so, somebody can raise their hand. But this is what I found. And I was actually shocked that it could fit, not only could fit on one page, but it could fit on one slide with the text not even made small, as we all do to stuff things in to a single slide. So this is what we've got in phase 2 and phase 3 right now. We have to do better. We have to do better. So there are many drugs that are waiting in the wings, and many of them you will hear about over the next couple of days. Unfortunately, some of them on this slide have already met their maker. But of these drugs, I think I come with a clear message, and somebody can stand up and say I'm wrong if you think I'm wrong. With 17 drugs already on the market, much of pharma, when I talk to them, are sending us a clear message. And the message is no business as usual. They cannot afford to spend millions of dollars and years of effort to develop a drug that is only marginally better than the ones that are currently on the market. It just isn't going to fly. Does anybody agree with me here? I know there are a few people who agree with me. So what does that mean? That means we have to figure out a way very early on to find out if a drug actually is better than what's out there now. And that is a tall order. Can we predict a better drug? A drug could differentiate itself in a number of important ways. It could be more tolerable for that 66%, or produce improvements in comorbidities. We would listen to that. We would want that. It could have specific efficacy in difficult syndromes, such as Dravet or Lennox-Gastaut. It could be anti-epileptogenic or disease-modifying, or of course it could treat those treatment-resistant. We are hoping for a way to predict any of these properties. It doesn't seem that hard, right? But matching human efficacy to animal models is something that we just aren't good at yet. We have many animal models of both treatment-sensitive and treatment-resistant epilepsy. We do have some models that mimic human disease. But if we're going to use these models to select the next best drug, we have to know how predictive the models are, both of overall efficacy, of specific efficacy in different syndromes, and also of tolerability. But unfortunately, at present, we don't have that capability. And ironically, we need to find a better drug to validate the model, because if all of our drugs are about the same, then how are we going to know, if we can't rank them in a model, that model will actually predict a better drug? This mouse looks about as confused as most of us are right now. So in order to say, do we have better drugs and worse drugs right now, we actually did a thought leader poll where we took very experienced adult and pediatric epileptologists and asked them to rate each anti-epileptic drug, both in efficacy and tolerability. And the original plan was to use that to compare with animal models. And on what I'm going to show you, if we thought that it didn't work, it was it was a red, and if it was very positive, it was green. And the interesting thing to me is people did it in different continents, in different states, in different houses, all by themselves, no groupthink, and they just sent their information back into the mothership. And a few people who did this are in the audience right now. And interestingly enough, there was very little variability, and in many cases, the results were unanimous. And these are the results, and I'm not going to belabor them other than to say that the sad fact is that the drug that got the best score was carbamazepine. Carbamazepine that's been around for 25 years. That is kind of sad, and it means that we have not found that drug that's going to differentiate itself yet. And just to show you, like a drug, tiagabine, that in randomized controlled trials seemed like it was a very good drug, in the clinic, the doctors didn't think it was very good at all. And just another point that I want to make to all of you developers out there, that if you look at the row for tonic-atonic seizures, there just was no drug that anybody thought was particularly effective. There's no green there at all. But so if there was a seizure type that you wanted to go for, this is information that might help you understand where the need is. Now we go to perceived adverse effects. Well, unfortunately, the same drug that people thought was the most efficacious, people also thought what had the most adverse effects, which is why carbamazepine is not number one in terms of the prescribing right now. But I want to focus on another drug for you, and that is lamotrigine. And I want all of you developers to pay close attention. Lamotrigine is the little drug that shouldn't but did, right? So if you look at the animal models, if you look at the original randomized controlled trials, it didn't look that much better than any of the other drugs in randomized controlled trials. And it has a serious adverse event of rash that can actually make you very, very sick. It has lots of drug-drug interactions. So why is this drug so popular? Why do people prescribe this drug with such regularity? And believe me, if there wasn't a slow titration, they would prescribe it even more. And I will tell you that, you know, this sounds like the beginning of a joke, but it's not. But 10 epileptologists were on their way to a meeting, and they were sitting in a car, and one said to the other, if you got epilepsy tomorrow, what drug would you put yourself on? And every single person in that car said lamotrigine. Every single person in that car. And I've asked that question repeatedly, and I always get the same answer. So why? There are drugs that are more potent, there are drugs that are easier, that don't have adverse effects. Why lamotrigine? Because of the adverse effects. So how can you predict another lamotrigine? We want another lamotrigine with no side effect, no titration requirements and no serious adverse effects. But unfortunately, we can't predict that. But I'm telling you guys, if you had that, that would be a value added. Value added can come in a lot of different ways. And we just have to find better ways to predict it. We have to find better ways to do early trials to be able to differentiate these drugs. So in summary, there are many new drugs in the pipeline. We need to get more drugs into phase 2. We will need to determine which of our animal models are predictive of better drugs. At the moment, we can't predict relative efficacy of drugs, and at the moment, we can't predict efficacy in severe epilepsy syndromes, and at the moment, we can't predict the next lamotrigine. But those are our challenges for the next few years. Thank you very much. And now it is my great pleasure to introduce my colleague, Bob Fisher from Stanford, who is a professor of neurology, and most of you know him. He has done so much for the epilepsy community. He's former AES president and is probably the most knowledgeable person on the planet about epilepsy devices, which he's going to talk about now.
Thank you. Having just said that I'm such an expert on devices, how does this device work? Here you got my talk. Good. So drugs are the mainstay of treatment for seizures and for epilepsy, and surgery is the way to go when it is potentially curative. But when neither of those are possible, then devices are becoming increasingly important, not as a cure, but as a potentially useful palliative therapy. And that is what I will talk about today, therapeutic devices. I live at ground zero of the startup street in the world, Sand Hill Road, and I hold many pieces of paper that may someday be worth something. So these are my conflicts. My main research has been with neurostimulation, and I don't take any support from Medtronic. My talk today will be an overview, kind of like a table of contents for the meeting that comes next. So let me start with seizure alert devices or seizure predictors. There's a watch, two watches, smartwatch and EpiAlert, that can pick up the shaking of a seizure, Bluetooth it to a cell phone, and then SMS message to mom or dad that Johnny is having a seizure, along with GPS information about where, because what could be worse than knowing your kid's having a seizure and not knowing where. This shake detector is useful to potentially bring aid to the bedside with a seizure, and you will hear later from Dr. Anu Nathan about this device. Cyberonics is about, I understand, to come out with a chest patch device that can pick up shake, that can detect EKG and other physiological parameters, and you will hear about this device later in the program. Electrodermal response used to be called galvanic skin response. I'm not sure why the name changed. I think maybe it kind of was getting a bad reputation from lie detector testing. This is not a lie detector device, but it does go up with some seizures and therefore becomes another potential monitoring technology. And Dr. Roz Picard of MIT is in the audience somewhere, and you will hear more from her about this interesting new potential monitoring method. Motor activity and muscles are the ultimate final common pathway for seizures, and patterns of seizures can be picked up in muscle. So you'll hear later from Dr. Jose Cavazos about the possibility of EMG, from surface EMG seizure detections. RTI International will tell you about a mobile seizure alert system. And so you see that there are many of these alert devices that are coming into play. Now, I'm not going to talk about diagnostic devices, but in some ways, a diagnostic device that could pick up seizures on the EEG would be part of a therapeutic loop. And yet the problem with that has been the difficulty in doing non-invasive long-term monitoring of EEGs. There are several technologies that are coming into play that might help that, and you'll hear about one in EEG Patch a little bit later in the program. Detecting seizures is good. Predicting seizures would be better, because then you would have the opportunity perhaps to take a pill, spray a drug in your nose, turn on a stimulator, or maybe just sit down. So how much different would it be for a person with epilepsy if they knew when their next seizure was going to occur? And this seeming fantasy is potentially coming true. The last slide, I'll go back to it because I didn't explain it. Implants subdural or epidural electrode leads, which then connect, much as, say, a vagus nerve stimulator would, to a subcutaneous device on the chest. That device monitors the EEG and broadcasts to a pager on your belt that shows colors. If the color is blue, you're in the clear. You're not going to have a seizure in the near future. If the color is red, please pull over your Mazda. And if it's white, it's intermediate. A trial by Mark Cook of Australia shows results with one patient. This is from his Lancet Neurology article of last year. Each line is a day in the life of one patient. The red arrows, there are 3 of them on the chart, and 1 in the legend, show that every seizure that occurred, which was at the red arrow, occurred during a time of red warnings. No seizures occurred when there was no red warning, although there were red warnings that did not have an associated seizure. So the blues were in the clear. I think this could change the paradigm of seizure management, because one of the worst things about having epilepsy is the uncertainty. And I hope this technology moves forward. Let's move now to adherence monitors. On the upper left, you see an example of a pill box that keeps track of every time you open your pills. On the bottom, you have a subcutaneous implant that broadcasts a readout of your drug levels continuously to a device. And on the right, those little green pills, if you think the NSA is spying on you now, wait until you encounter these green pills that broadcast a signal when they are dissolved by your stomach acid, indicating not only did you put them in the mouth, but you swallowed them. So let's move next to clinical information systems. I think this is likely to be one of the most important advances for taking care of people with epilepsy. The old-timers in the room will remember the great Dr. Kiffin Penry, who basically founded the NIH epilepsy branch. He taught us that paying attention to the details of when people have seizures, when they take their medicines, can enable you to customize your treatment for them and vastly improve the outcome. We need to apply today's technology to that message, because the biggest problem I have in caring for my patients in clinic is lousy information. I will put a patient on a new drug. They'll come back 3 months later. In fact, if we'd been keeping track, their seizures would have been cut in half, which would be a home run in any drug trial. But I say, how are you doing? They go, oh, I had a big one in the parking lot last Saturday. And that's the information I get. I don't know the drug is working. We have to be able to do better than that. And I won't even get into, with Jackie sitting on my left, the quality of information in traditional drug trials. She's laughing, but she's really crying. So we currently have 3 online epilepsy diaries, potentially more being discussed. They're electronic. Some of them, for example, my epilepsy diary, are used by as many as 25,000 people. So here is how I think the possible future will take place. The caption of the cartoon says, we're going to run a few tests to pin down the cause of your seizures. I modified that cartoon from T. McCracken. You'll have a person who has epilepsy. They will have some type of automatic seizure tracking device, and they will have some type of automatic adherence tracking, which will monitor when they take the pills and what the levels are in their system. This will go to an internet cloud, hopefully securely and privately, and it will give customized feedback back to the patient about what to do to keep them in balance. So we have the information we need to treat them, and we have real-time feedback to get the drugs or the therapies where they ought to be. I think this is the way that the management of chronic disease is moving in several areas, and epilepsy is behind some of the diabetes and hypertension advances in this area. We move on to optical control. Bacteria and hostile environments, such as the Dead Sea, have to pump salt out of the cell, and they use light to do that. Dr. Carl Diceroth at Stanford has developed a new technique called optogenetics to infuse the genes to do this into brain cells. You can make specific populations of brain cells sensitive to different colors of light and instantly turn them on and off with fiber optic light, as you can see with the orange bar inhibiting the cell. This is a potentially exciting way to stop seizures by inhibiting cells instantly with light, but it is invasive, involving viral transfection of brain and implantation of optical fibers. New ways of drug delivery. Here in San Francisco, if it's flammable, we will smoke it. You're going to hear later about the possibility of smoking your seizure medications from Dr. Casella, and based on a technique developed by our own Dr. Michael Rogowski, who's in the audience. Some years ago, I developed or used an animal model of epilepsy to show that seizures could be automatically detected from the EEG, and a programmable infusion pump could be instructed by the program to deliver a squirt of an anti-epileptic drug, in this case diazepam. Please don't put diazepam in human brain. It was just an experiment. And you can see in the lower right corner how the diazepam shut off the seizure. There are experiments underway to do the groundwork for clinical trials of brain infusions of medications. Again, it's quite invasive, but the advantage is potential achievement of a very high therapeutic ratio at the place you need it, and you also may be able to deliver drugs that otherwise could not pass the blood-brain barrier. The next issue is focal energy delivery. I think the first of this was radiosurgery. This is a CyberKnife machine developed at Stanford. You may gather I'm a Stanford partisan. And that beam swings around the head in a computer-controlled way, painting the radiation dose on the brain according to how you've painted the MRI on the patient ahead of time, and it only delivers the radiation dose in a crossed beam to the area that you want to deliver. So a multi-center prospective trial, the ROSE trial, showed that stereotactic radiosurgery was about as effective as open craniotomy surgery. And it's nice because you don't even have to cut the hair to do this treatment, but the problem is potential long-term effects of radiation. How often my patients have asked me at a time of discussing seizure surgery, "Doctor, can I have this surgery done with a laser?" And for 28 years, I've said no. So now you will hear from Bill Hoffman that the answer has become yes, and you'll learn about this new therapeutic technology. The newest kid on the block is focused ultrasound. This is in advance of being used for epilepsy surgery, but it is possible with new phased arrays of ultrasounds directed to an individual map of a person's brain to get very precise delivery of high-energy ultrasound to certain points in the brain. And although it's not been used for epilepsy surgery, it's been used in patients for tremor and starting to be used in other conditions. And this doesn't have the radiation effects long-term. So you will hear later from Dr. John Stern about focused ultrasound as a very new treatment, potentially for seizures. So we come at the end to neurostimulation. Neurostimulation in general is mildly invasive, I think, but it doesn't require resection of tissue. It's adjustable and it's reversible. Those are its advantages. The original neurostimulation was vagus nerve stimulation. There is a new generation of devices coming out that will deliver a stimulus to the vagus nerve based on detection of ictal tachycardia. There have been trials in Europe and in the US that have just completed, and Dr. Paul Bone and I will be presenting the results of those trials at the next AES meeting. Magnetic and electrical brain stimulation is attractive because it's non-invasive. You just come in and you have a few treatments each day. Half of the trials have shown efficacy in seizures and half have not. And my sense is that this depends upon whether you know where the focus is, whether you can get right over it, whether it's superficial. Dr. Chris Giorgio developed trigeminal nerve stimulation as a technique. The definitive trials are not done, but the preliminary trials are promising. It's much like vagus nerve stimulation conceptually, except it's done on a branch of the trigeminal nerve around the eye, where it's accessible by paste-on electrodes rather than surgical electrodes, so you can try it non-invasively for some months and see how it works. Last is brain stimulation. There have been two pivotal trials showing efficacy of brain stimulation for epilepsy. I won't read this table in detail. The one on the left is responsive neurostimulation, led by Dr. Martha Morell and sponsored by NeuroPace, and you will hear later from Dr. Frank Fisher, I think, more about this trial. It detects a seizure based on customized recognition of that patient's EEG seizure pattern and then delivers stimulation to counteract the seizure. The column on the right is thalamic deep brain stimulation, the so-called Sante trial, sponsored by Medtronic, and I was the first author of the publication on that trial. It has about the same efficacy, which is in the range of about a 40% median reduction of seizures compared to baseline, as does responsive neurostimulation. My view is that the results of both of these are very similar, that they will be complementary to each other, although responsive neurostimulation mostly applies to situations where you know where the seizure focus or foci are. RNS is FDA approved in the United States. Discussions continue with the FDA on thalamic DBS, but thalamic DBS is approved in over 30 countries internationally. So what comes next from this? Where are we going? We're going backwards is where we're going. I'll close with this quote attributed to John Galsworthy, Woody Allen, and Yogi Berra at different times. We can predict everything except the future, except I think the future is going to be better. Thank you very much.
I just want to know, do you think it's going to be better for everything or just for epilepsy devices? So I know we're running a couple minutes behind, but I want to give an opportunity for just one or two questions, if there are any burning ones. And seeing none, I am going to move forward with the next session. So thank you, guys. So it is my great pleasure to start out by introducing Rusty Katz, who is the former, he's now, his job title here is Consultant Epilepsy Study Consortium, but many of us know him from his former employment at the FDA, where he was the division director for neuropharmacology for many, many years. He'll probably tell us how many. And he will also tell us that he can tell us about the past, but he cannot predict the future. Right, Rusty?
Thanks, Jackie. Yeah, let's get that out of the way, right. I can't predict the future. But it's a pleasure to be here. Although it is a little sobering to think that now, after I reached a certain age, every talk I give starts with the word past. So, but actually it's reasonable, at least in this case, because almost everything else in the title of this talk is misleading. I don't work at the FDA anymore, and I can't speak for the FDA anymore. We've already heard eminent people have said that we can't predict the future. I will talk a little bit about the present, only in the sense of initiatives that are being worked on by various people, not me, that hopefully will affect the future. I'll speak a little bit about the past, and also what's misleading about this title is that it says epilepsy trials, and the initiatives I'm going to be speaking about have as their goal the fact that trials won't be necessary anymore for approval in certain restricted conditions. So, with one exception. So, I don't want to lose sight of the fact, and I'll say it at the opening, that what is being anticipated, what is being proposed, will represent, if it's adopted by the FDA, a radical departure from how things have been done in the past. That may be a good thing. And again, I can't predict that the FDA will adopt these, although these initiatives are being done with the knowledge of the FDA and in some cases with the participation of the FDA. So they're well aware of what's happening. But the goal, again, in most cases is to no longer require clinical trials for certain specific settings. So let's just talk about what those initiatives are. And my goal here is to present these initiatives a little bit, I think, about where they are, although I am not directly involved in any of them. I was involved in some of them when I was at the FDA. Not to go into excruciating detail about how these things are evolving, what the results are. I don't know what the results are in most cases, and the work is still ongoing on most, if not all, of these initiatives. But the idea is to just give you a little taste of what's actually happening and what the potential is for how drugs might be approved in certain settings in a very different way from how they've been approved in the past. So that's really the idea. So there are really, I don't know if it's cut off on yours. It looks cut off on the left over here. I don't know why that is. Anyway, there are basically 3 initiatives that I want to talk a little bit about, and if we have some time, I'll talk about the last one. So work is being done on looking at new ways to approve drugs for monotherapy or to grant monotherapy claims for drugs, again, without doing monotherapy studies. So look at how drugs might be approved in pediatrics, again, with the goal of not having to perform controlled trials in pediatric populations in order to get those approvals. There is work being done on new designs for add-on trials for very specific reasons, and we'll talk about that. And then finally, again, if we have time, a recent precedent for the approval of a controlled release product that also did not require the conduct of a controlled trial. And again, that was in the Neurology division. That was a pretty radical move, although a perfectly reasonable one in this case. So I'll just go through these one by one and just give you, again, a taste of the approaches being taken and the reasons why these new approaches are being undertaken. I can see they're all cut off. All right, so let's start with monotherapy trials. So historically, monotherapy studies, as you probably all know, have been difficult to do. There have been many designs that have not just been proposed, but actually been undertaken to attempt to demonstrate that drugs are effective as monotherapy. As you know, drugs anticonvulsants are routinely approved on the basis of so-called add-on trials, where their drug is added on to background antiepileptics, or placebo is added on to epileptics, and that's how effectiveness is established. So anyway, previous designs have included very short-term studies in inpatients. These are on the order of several days. These studies have been problematic from the point of view of interpretation, or at least generalizability, because drugs, of course, are given chronically, and it's hard to know what a 2 or 3 day study predicts for the use of a drug as monotherapy in the long term. These have been usually in pre-surgical patients who had their drugs removed anyway to evaluate them for surgery. Studies have been attempted and done in newly diagnosed patients. These studies are difficult to do as well. In the past, low dose versus high dose withdrawal to monotherapy studies have been done. These studies all have, again, difficulties in their interpretation, as well as posing ethical problems. For example, the many low-dose versus high-dose withdrawal to monotherapy studies were done until people began to realize that, well, randomizing patients in the monotherapy setting to a low dose, not a maximally effective dose of a drug in question or a comparator drug, really isn't that much ethically different from randomizing patients to placebo, and nobody thinks randomizing patients to placebo, chronic patients anyway, to placebo in the monotherapy set in epilepsy is a good idea. Recently, historical controlled withdrawal to monotherapy studies have been utilized in a couple of cases. That is to say, these are designs in which patients are withdrawn to monotherapy, but there is no concurrent control group, and their responses are compared to historical data gathered from these low-dose versus high-dose withdrawal studies. It's a clever idea. The agency has relied on these studies in a couple of cases to approve drugs as monotherapy, but there are difficulties with those studies as well. You are randomizing patients, and they're potentially getting worse in the trial. It's really just an attempt to show that the drug is better than the historical placebo or low-dose groups. So all of these designs are potentially problematic from the point of view of concluding that drugs work as monotherapy. And of course, it bears repeating. As I said, drugs are approved as anticonvulsants are typically approved as add-on. And the label says that they are add-on, and that's the source of the problem. From the point of view of monotherapy, it is certainly not necessary for approval, obviously, to get a monotherapy claim. But there has been, over the years, and increasingly, there's great interest in obtaining monotherapy claims. For a number of reasons, folks feel that there at least are groups of physicians out there who won't treat with monotherapy, which is, of course, the desired situation, unless there are specific labeling indications for it or dosing for it. Insurance might not cover the use of monotherapy if a drug is only approved as add-on therapy, even though physicians will use it as monotherapy. So there are a number of reasons that folks want monotherapy claims. As I say, the agency hasn't been willing, at least to date, to grant a monotherapy claim based solely on add-on studies for several reasons. A simple reason is, and a historical reason, is that the agency has felt that the claim granted ought to be based on the studies that were done to support approval. And since, in most cases, the studies were done only in the add-on setting, it made sense to say, well, look, the add-on studies were done. That's the claim that ought to be granted. This actually, this is a part of the past, the part of the past that I still hold on to the memory of. There were advisory committee meetings in the early 90s that discussed the first 3 drugs of the sort of the new generation, and 3 days of advisory committee meetings. And the principle was articulated by the committee at that time that the claim ought to be based very specifically on the studies that were done. So that's why, and that principle has held forth to date. So these drugs are approved as add-on therapy in adults with certain types of seizures, the seizure types that were studied, usually partial seizures at the outset. So that's historically how the current claims came to be. And there were questions raised about whether it's reasonable to extrapolate effectiveness as monotherapy when a drug has been shown to work only in the setting of background AEDs as add-on. And even if we could extrapolate effectiveness, how would we choose the dose? How do we know that the dose that is effective as monotherapy is derivable from the dosing information that are obtained in studies done when the drug was used as an add-on? So this is the historical case, and these are the issues that the agency has been pondering about extrapolating the effectiveness as monotherapy from add-on studies. So nonetheless, the agency has been willing to consider the field's eagerness to obtain monotherapy claims from data generated in add-on studies. So in this regard, many of you probably know that a working group of academic and industry experts is producing a white paper that it's far along, as far as I know, it's not finished, that attempts to address these concerns. And I would just point out that this white paper approach, which I think has been really used to great effect by Jackie French and some of her colleagues, has worked in the past, and I think it's a useful way to try to get the agency to move on certain issues. it was this sort of consensus white paper approach that led the agency to agree to this historical control withdrawal to monotherapy design that we talked about before. So it is a useful mechanism, I think, to approach the agency with. Anyway, I'm not going to go into great detail about the specific arguments. It's a detailed document that goes in great depth in a number of issues. But I will just say a few words about some of the sort of the highlights of the argument. One argument that the white paper being generated makes is that there has been considerable inconsistency at the neurology division in the past about claims granted to drugs. So, for example, a drug might be studied in multiple sclerosis as monotherapy, yet the claim is granted for the treatment of relapsing remitting multiple sclerosis, or a drug might be studied in Alzheimer's in a certain setting, but there's no specific add-on claim for Alzheimer's drugs or monotherapy claim for Alzheimer's drugs. These drugs are approved for the treatment of dementia of Alzheimer's disease, and on and on. So that claim has merit. It's not always true. Sometimes in some of these other settings, the neurology division has granted specific claims as add-on or monotherapy using a different language, but that's the implication. But largely, I think, having had been at the FDA for 30 years, I think I'm in a position to say that there has been inconsistency. So I think that's a fair point that needs to be considered. Other arguments that the white paper makes is that their non-clinical data non-clinical data for all the anticonvulsants coming down the pike certainly suggests that in each case the sole agent is effective. The non-clinical data typically generated in these so-called monotherapy state. So there's very good reason to believe, based on preclinical data, that in fact these drugs work by themselves and don't need other anticonvulsants behind them. There's the case that most previous studies of drugs done in monotherapy have been positive, so that whenever a drug is studied as monotherapy that had been previously studied as an add-on have been positive. Not every study, but most. And although it is true that there isn't a huge background of adequately designed monotherapy studies, as there have been for add-on studies. So, but nonetheless, this is an argument that the paper makes. Another argument that the white paper makes is that clinical practice suggests that drugs that have been shown to be effective as add-on, in fact, are effective when they are used in patients as monotherapy. This is not what I would call data, but it is an impression, certainly, that many folks have. And also, it makes the case that there's no data to support the conclusion that a specific background anticonvulsant or combination of anticonvulsants are necessarily necessary for a new anticonvulsant to be effective as adjunctive therapy. In other words, these drugs, when they're approved, are added on to many different combinations of many different anticonvulsants. And the fact that they are shown to be effective as adjunctive therapy in that setting does strongly suggest, according to the paper, that in fact, they must work by themselves. The agency doesn't, and this is a point the paper makes, doesn't in particular go back and analyze add-on studies by looking at every other specific combination of background anticonvulsants that the new drug has been added to. So the fact that it works in the setting of many different types of combinations does suggest that the drugs have intrinsic anticonvulsant activity by themselves. The case is made that adverse reactions seem to be less in monotherapy compared to when drugs are added on to other background anticonvulsants. And the case is also made that in dosing, which is a question I raised before, how do we know what dose to use in monotherapy if the drugs have only been studied as against the background of other anticonvulsants? This white paper tries to make the case that in trials when monotherapy trials have been done, and again, there haven't been that many, but when they have been done, the effective dose range more or less overlaps the effective dose range when it's given as adjunctive therapy. So these are, in broad brushstrokes, the arguments or some of the arguments that the paper makes. I would point out, though, it is possible due to pharmacokinetic interactions that the plasma levels of an anticonvulsant in monotherapy may be greater than when they're given as adjunctive therapy, depending upon the potential interactions. So it is possible that you do need a higher dose in monotherapy than has been studied or been shown to be effective when it's given as adjunctive therapy. So this is something that I think the paper will attempt to address. But nonetheless, these are the arguments that are laid out. And certainly the FDA is aware of ongoing efforts to try to extrapolate to monotherapy from adjunctive studies. So that's one major initiative that is ongoing. Another major initiative that's ongoing is pediatrics. The agency has, to date anyway, always required a clinical trial in pediatric patients in order for a pediatric claim to be granted. These drugs typically, again, are approved based on studies in adults only. That's actually fairly unfortunate. The agency has regulations that allow for a pediatric claim to be based on data in adults if it is determined that the disease in question is sufficiently similar between adults and pediatric patients, and pharmacokinetics and pharmacodynamics are sufficiently similar. The agency also requires that studies be done in children for drugs that are approved in adults if the indication that it's approved for in adults exists in children, and obviously that's the case here. I said it's unfortunate before that pediatric studies typically in drug development programs are done quite late. In fact, they're often not undertaken until the drug is already approved in adults. That's unfortunate. The regulations don't require that, but companies tend to do these studies later and later and later to the point where it can be very, very difficult to do pediatric studies once a drug is approved for adults because the drug is available. And why would you enroll your child in a study where there's a possibility that they might get placebo if they can just get the drug? So I think that's the fact that there haven't been many controlled trials of pediatric patients is an artifact, in part, about how drugs have been developed for pediatrics, and I think that has been unfortunate. Anyway, the agency's never granted a claim for pediatric epilepsy in the absence of a controlled trial in pediatric patients because they haven't felt, we didn't feel, and they haven't felt that the criteria of similar pharmacodynamics and disease similarity have been adequately addressed. Nonetheless, obviously, people want information about whether drugs are effective in pediatric patients. They want dosing information, even though the drug's only been studied in adults. And so there has been renewed interest in trying to address the question of how can we get to a pediatric claim without doing studies in pediatric patients. Again, another working group has been impaneled consisting of industry experts, academic experts, and also enlisting the aid of pharmacometricians at the University of Maryland. And the idea here, it's a two-pronged approach that they're taking. One is to attempt to establish that the disease in pediatric patients is the same as it is in adults. And also to show that where there have been studies in pediatric patients, that the effective plasma range in adults is the same as the effective plasma range in pediatric patients. So these are attempts that are being undertaken. This pharmacokinetic approach is very analogous to an approach we took in several cases when we approved a drug for pediatric monotherapy, where there were studies in pediatric add-on studies and adult add-on studies and adult monotherapy studies. In a couple of drugs, we said, well, if the effective plasma range in add-on studies for pediatric patients is the same as it is for adults in add-on studies, we will assume that the effective range in pediatric patients as monotherapy will be the same as the effective range as monotherapy in adults. And we just extrapolated to approval in pediatric monotherapy. And the approach that's being taken now is a direct extension, in my view, of those analyses which have been accepted. It's not entirely obvious from a kinetic or mathematical point of view, how do we establish that the effective the effective plasma range is the same? What do we mean by same? How do we test to say, well, these are the same in adults and pediatric patients? But nonetheless, it's a tractable problem, and it's been solved in the past. All right, and so if it's true that in every case that they're looking at, the effective plasma range in pediatric patients as add-on is the same as the effective plasma range in adults as add-on, it might be reasonable to say, well, all right, it's always true. For the next drug that we won't study, we expect that it will be true that the effective plasma range in pediatrics will be the same for the next drug as it is in adults. So that therefore, the contention is we would be able to extrapolate to pediatrics from adult data. Safety, of course, would have to still be assessed in pediatric patients. That's not the kind of thing that is easily extrapolatable. The best type of safety data comes from controlled trials, and if we don't do controlled trials in pediatric patients, then it's going to hard to be it'll be hard to know how to evaluate the safety data. But nonetheless, it's a possibility. And again, this white paper that is being generated by this group will also address the similarity of the disease in both populations, largely based on analogies from rodent to human. But the paper makes great efforts to establish the similarity of the disease. So that's another initiative. The last initiative I want to talk about is the design of add-on trials. You know, these studies have typically been 3 months duration, maybe a little longer in some cases. It's been considered a perfectly ethical design. They're always done because you're adding placebo on to some people, but against the background of their standard drugs. So that's been considered perfectly ethical. However, you're probably aware of a publication by Rivlin and his colleagues a few years ago, which looked at a meta-analysis of 112 controlled trials. This concluded that the risk of SUDEP was actually markedly less in patients who got the drugs added on to background compared to patients who had placebo added on. So that raised serious questions about the ethicality of even that study design, which has been the standard design in which drugs have been approved since at least the early 90s, if not before. So in the worst case, if this turns out to be true, you could imagine that this would mean the end of placebo-controlled add-on studies, and that, of course, raises the very, well, I'll say scary question of to be replaced by what? If we can't do adequately controlled trials, where do we go from here? In any event, in light of the findings, the agency took this very seriously, and the agency can't act, I don't think, in most cases based on a literature report. So the agency has been undertaking its own analysis of controlled trials. This is a very time-consuming operation, asking companies to submit the data. Of course, the agency has knowledge of trials that aren't necessarily published, which is where Rivlin got his data. So this effort, as I understand it, is getting very close to being finished. And so the agency has been looking at this question to see whether or not it can confirm the findings that Rivlin found. And so that's ongoing. But this, of course, has spurred to the, let's assume for argument's sake, the agency agrees that, yes, in fact, patients on placebo in add-on studies have a higher incidence of SUDEP than the patients who actually were randomized to the active drug. What do we do now? Well, Jackie again and some of her colleagues have been looking at the question of looking at new endpoints with an idea of minimizing the period of time that the patients will be exposed to placebo, not doing away with it, but minimizing it in the hopes that that would help address the question of SUDEP. And these approaches are looking at the various variants of, instead of just looking at a 3-month treatment period, looking at, well, what's the time to the nth seizure, the 4th seizure, the 5th seizure once the study starts, in the hopes that this would shorten it. And one thing it's particularly emphasizing is looking at a new outcome, which would be the time that the patient reaches their baseline seizure rate. So if they had 10 seizures per month in baseline, they would look at the time for the patient to reach their 10th seizure, with certain minimum amounts of time and maximum amounts of time in the study. So they are comparing the results of this new outcome with the results of the traditional outcome in previous studies that were done as add-on. I think they're looking at maybe 3 drugs with a total of 12 studies, at least that's the last version I saw. So it's important to compare the old outcomes with the new outcomes to see how they compare, especially if the agency is considering changing to this new design. These efforts are being discussed with agency staff. The agency is very well aware of what's going on here. And then I'll have to look at safety data too. If we have 3 months of controlled trial safety data now, how will we interpret the safety data from these much shorter studies? So these are questions that will have to be addressed. Finally, I think I had a half hour, so I don't know if am I
Yeah, you take about 2 more minutes.
2 minutes, okay, anyway. Finally, I just want to present this most recent case, a controlled release products, very common. We always made companies do controlled studies of controlled release products because we weren't really sure what the shape of the plasma time concentration curve, whether or not that was important for effectiveness. Companies have suggested, well, let's just compare Cmax and the area under the curve of the controlled compared to the immediate release and say if those are equal, then that should show that the controlled release product is effective. We've never, the agency has never bought that. You can see here, if you think Cmax is important, it doesn't make any pharmacologic sense to me to say, well, yes, Cmax is important pharmacologically, so if we only achieve it once a day with the controlled release, that should be good enough. I don't know what the pharmacologic basis for such an assertion would be. So the agency has never, because these curves look obviously very different, we've said if the curves look different, you have to do a controlled trial. Okay, I'm going to just skip this. But anyway, there is, recently the agency has approved the topiramate control release product, which is given once a day, and it was compared to twice a day. And it didn't just meet the usual, the Cmax is the same in control release and immediate release, and the area under the curve is the same. It met the bioequivalence standards for Cmax, not just at Cmax, but at every time point. So the plasma levels were equivalent at 2 hours, at 3 hours, at 4 hours. I don't know when Cmax was, but throughout the 24-hour dosing interval, except for the first 1 or 2 hours, the plasma levels were always the same, given by the sort of generic equivalence standards. And not only that, the area under the curve was not only the same for 24 hours, but it was the same from hours 0 to 2, hours 0 to 5, hours 0 to 8. Wherever you looked at a partial AOC, they were always the same. So even though these two curves looked different, they looked almost a little like this when you just looked at them. At each time point, the plasma levels were actually the same, and these partial AOCs were also always the same. So the agency considered that perfectly reasonable to establish that these drugs would be therapeutically equivalent. So that's an example where a controlled release product was approved without a controlled trial. And that's a very rare thing. So let me just finish. There are numerous efforts being undertaken to establish new forms of acceptable data in several important areas in epilepsy, in monotherapy, in pediatrics, and shortening studies in add-on designs. The agency is absolutely open to these new approaches. We started them when I was there. They're ongoing. Many of them are coming, I think, very close to being finished, and the agency will presumably rule on them. And the agency actually has been involved in most, if not all, of these efforts. So they're very eager to look at these. Again, as I said at the beginning, we shouldn't lose sight of the fact that this is a radical departure, or would be if it's adopted, a radical departure from how the agency has done business before, which is do a clinical trial, get the empirical result, and grant a claim based on it. Here we're talking about granting claims in cases where no data have been generated in that specific setting. We shouldn't do that lightly. We should also recognize it's a relatively weak epistemological or philosophical position. In other words, we're saying, well, we have some data, and in the data we have, it looks like pediatric dosing is the same as adult dosing, let's say. But we don't have a wealth of data comparing these situations, and we're saying, well, it worked in the past, therefore we concluded will work in the future. That's not a given, and that's a complicated position to take. Nonetheless, it may be very reasonable in these cases, and I'd say just stay tuned. I gather the agency will be making some decisions about this these issues, hopefully in the relatively near future, and they certainly seem to be favorably disposed to doing it. There's a meeting in Stockholm next month where these initiatives will be discussed in a meeting with the EMA, and there will be FDA representation at that meeting. So I hope we will learn more about where these things are headed in a month. I think I'll stop there.
I just want to say, as Rusty steps down, that the tone that he set of collaboration with the community is really why these efforts can go forward, and it has set the tone for the next administration. And I think we owe him a great deal of debt. We gave him an award for exceptional contribution at the American Epilepsy Society last year, and he was well deserving of it. So a round of applause to Rusty. Okay, so we are going to switch gears now, and we're going to talk about. I failed to mention that this group of talks is called "Why Is It Better in 2014 Than It Was in 2013 or 2012?" We're trying to start off on a very positive note. So you just heard from the FDA why things might actually be better moving forward, even though we can't predict the future, that there are some of these exciting initiatives going on. And next, I am very happy to introduce my friend and colleague, Chris Gallen, who is the president and CEO of SK Biopharmaceuticals. They do have an epilepsy drug in development, and he's going to tell you why people should continue to invest in epilepsy.
Good morning. Why should industry keep investing in epilepsy? The short answer is because we can win, because we can succeed, because it's possible to create better therapeutics. Now, I'd like to begin looking at why I think that's true, sort of in the big picture context. From the Neolithic period, 8,000 years ago, when agriculture was invented and civilization began, all the way through to the late 1700s, there was basically never a society in all of human history where the average man or average woman lived past the age of 35. Life was, in the phrase of Hobbes, nasty, brutish, and short. This began to change around 1800. 1798, Jenner invented the smallpox vaccine, whose successors eventually eliminated smallpox from the face of the earth. 1806, morphine, one of the first small molecule therapeutics, was isolated. In the ensuing years, the combination of German organic chemistry, originally invented to make prettier dyes for clothing in an affluent bourgeois German society that was emerging, were applied to natural products being isolated in England, Italy, and France to produce the first pharmaceuticals. At that time, in 1800, average lifespan in the United States and the South was about 20. In the North, it was about 25. By 1900, it was 50. By 1935, it was 65. That's why the retirement age for Social Security is 65. They figured half of the people they'd never have to pay off. Now it's about 82. We've made life longer. We've made life better. We've removed a lot of suffering from the world, but there is so much more to do. The combination of academicians, physicians, advocates, public health workers, and the whole therapeutics industry, small molecules, biologicals, vaccines, diagnostics, in the future, genetic therapies. Together, we've changed the world. This is the fundamental substrate for why the industry exists. In the course of this, we've created a $1 trillion therapeutics industry and a $10 trillion healthcare industry. Why epilepsy? First, why CNS? Since CNS has been an area that companies have been abandoning. When you look at any disease to figure out how important is it in its effect on the real world, one thing you could look at is how many scripts are written for medicines in that area. What you can see is that more scripts by far in the blue are written for CNS drugs than any other therapeutic area. It's by far the largest area of unmet need. I might note parenthetically that that little purple triangle, oncology, is a small number of scripts, but it happens to be a large dollar value area because pricing is so high and because approval is so much easier. Why do companies not invest in CNS? Why have they abandoned it? There's one big reason, because the success rate in CNS therapeutics, seen there bringing up the rear on the right of the chart, are appallingly low, 4 to 5%. Why is that? And why is an area, for example, like infectious disease, or if we put the curve up here for oncology, very similar to infectious disease, why are other areas experiencing much higher levels of success? And what does this have to do with epilepsy? Well, I'll talk briefly about why infectious disease has such a higher rate of success, and note that the conditions for success in infectious disease, while they're not true in central nervous system diseases in general, do apply in certain central nervous system diseases, and I believe epilepsy is, in part, one of them. The big difference between, from my point of view, easy areas like infectious disease and oncology, and I can say nothing is easy, but having been involved in registering infectious disease products and oncology products, I have some sense of it, is they have two things going for them. First, they're dealing with a simple system. You can grow infectious disease in a dish. You can grow cancer in a dish. You can study it in a cell. And they have a high level of predictivity. If an anti-infective works in a test tube, and if you can deliver it in human beings at the right concentrations to the right tissues at the right time, it's basically, and if the patient lives through it, if they tolerate it, it's basically a pretty good chance that it will work as a therapeutic. Central nervous system, not so simple. 100 billion neurons, using over 100 different neurotransmitters, maintaining 100 trillion connections, firing basically multiple times a second, all arranged in complex systems that modulate each other, that interact with each other, and that often have in their complexity a compensatory mechanism such that when you poke the CNS with one therapeutic hitting one part of the system, the CNS pokes back. The CNS tends to compensate and neutralize the changes that you've made. Complicated system. It's hard to, in that situation, while you can, in oncology or infectious disease, pretty much understand from the initial genetic cause to the final consequences, the complete sequence of how it is, the logic of the disease process, the pathophysiology, and you can figure out a targeted intervention to block it. This is virtually impossible in the CNS. It's just too complicated. There are exceptions to this, but there aren't many. So we deal with the fact that we deal with a more complicated world. The second thing is that for many central nervous system diseases, animal model predictivity is terrible. Why is that? Well, why should animal models matter? Ideally, the animal would be experiencing the same pathophysiology. You do it, completely understand the pathophysiology of the animal. You completely understand the pathophysiology of the human disease. And the two of these would be congruent in all important respects. The reality of it is that we rarely reach any of these conditions. We don't really understand the pathophysiology of the animals. We rarely understand the pathophysiology of the human diseases. And human beings, because they're outbred, are genetically very much more complex than our inbred mice, are rarely the same. And the two of these, the animal models and the human disease, are rarely congruent. They're rarely the same. You know, what is a schizophrenic rat? Does a rat have enough brain to hallucinate? Epilepsy, in the grand scheme of things, is not perfect, but it's not the job of industry to succeed in a perfect world. It's the job of industry to figure out how to succeed in the real world. And while I think we can argue that epilepsy models are imperfect, that our understanding of the disease is imperfect, I'm here to say that compared to developing an antidepressant, epilepsy models are perfectly predictive. They're great, right? This is true for many different illnesses. Epilepsy is not simple. Not only are there many mechanisms involved, but from the excellent work of Jeffrey Nobles characterizing the detailed genetics of epilepsy models, you begin to realize that even in highly inbred animals with identical behavioral syndrome and identical EEG characteristics, you can find several different combinations of the same sets of neurotransmitters that will all produce the same phenotype, the same expression. I think from trying to understand the epilepsy from the fundamental level is not, it's not in the cards at this point in time. It's just too complex. But we do have models that are pretty predictive. We've devoted a great deal of energy to refining those models, improving those models, and developing a robust array of them. What we found is that many animal models have been developed that look at individual strains of pathophysiology that happen to show up in one or another patient, one or another epilepsy. And when we isolate molecules that are very robust against the range of models, in general, they have a pretty good chance of actually succeeding in human disease. We're not looking for 100%. We would be happy with 30% in terms of success rate, because the current success rates are more like 5% across CNS, although we think higher for epilepsy. What we find as we screen our molecules, looking for molecules that are very robust and very powerful, and we'll give an example of one tomorrow that we think has a lot of potential, is that rarely do they work through one mechanism. Almost every molecule that we find that's really promising, that's really good, hits 2 or 3 mechanisms at least. Perhaps more than that, 2 or 3 that we have been able to discover on the molecules. So what we do is to develop the chemistry to manipulate each of those mechanisms relative to one another in order to optimize the activity of the molecule. And again, you'll see a result tomorrow. So why should we invest in epilepsy? First, because it's doable. It's possible to succeed. Epilepsy is an area where the animal models have a reasonable positive predictive validity, where there are some human screening models, for example, the photoconvulsive epilepsy, that has reasonable predictive capacity, and where it's pretty straightforward to measure whether your drug is working or not. We were talking about technology earlier this morning. No doubt, objectively being able to count the seizures would be even better than relying on diaries. But I'm here to say that relying on diaries of counts of events is a hell of a lot more accurate than asking someone, how do you feel today? As a measure of efficacy of your drug. And because while there is a placebo effect in epilepsy, and I think it's a placebo effect that reflects a great deal that people become more compliant when they're in the clinical trial, the placebo effect itself is a manageable level. It's a reasonable level, about 20%. Why invest in epilepsy? Because it's a great market. It's a serious illness that has considerable unmet medical need. There's 3 million patients, about 1% of the population. It's a $4 billion market growing to $5 billion, and spending a lot of my life in Asia, an area that is growing phenomenally, that will add 3 billion population over the next 20 years, and where incomes of people are doubling every several years because they've adopted capitalism and they're just moving forward and focused on growth. The market's going to grow more than current estimates as the Asian middle class moves to the fore. Because specialty marketing is efficient. A small number of physicians, neurologists, well-trained, tend to dominate the vast majority of prescriptions, so you don't waste huge amounts of money paying for large sales forces. There's lots of challenges. This is a crowded marketplace with a lot of pricing pressure, a lot of generic erosion, which increases the pricing pressure. It's hard times if you're a Me Too molecule. As we talked about earlier, Me Too molecules probably not worth developing, even though they may actually be useful in a specific patient. But the seriousness and the disabling nature of the illness that we're treating is such that a differentiated, better medicine is going to command a significant market. Why invest in epilepsy? Because when you develop a drug that works in epilepsy, you are modulating one of the underlying most important mechanisms of brain functions, neurotransmission, neuromodulation. And if you develop a drug that affects neurotransmission and neuromodulation, it's going to affect other illnesses. Many of them can be bigger illnesses, bigger markets: migraine, neuropathic pain, fibromyalgia, generalized anxiety disorder, mood disorders. But they're bigger areas, but they're hard areas to conduct trials in. They're inefficient areas to conduct trials in compared to epilepsy. So as I see it, the logic of winning is all about your strategy. One of the fundamental elements of strategy in war and in business is to attack where your opponent is weak, to attack where your opponent is absent. The very fact that companies have abandoned CNS to a very significant degree makes an opening for companies like ours to penetrate this area. You then focus on picking indications where the cellular and animal models have positive predictive validity. Epilepsy is a good example. You then pick an indication where if the drug works, you pick an indication that is serious so that you don't spend your life at the FDA being nitpicked over all kinds of tiny safety risks because you're focused on dealing with a serious and major illness. So you can really define the safety profile of your compound. You then focus on an indication where if the pharmaceutical works, the clinical trial will work. In depression, half of the drugs that are approved when they're put in clinical trials fail in their trials. The drug works, but the trial fails. In epilepsy, for the most part, if a trial is well conducted, the drug works, the trial will work. So as I see it, the strategy is your first development and registration program should be focused on epilepsy. You use that to determine the right usage, the right dosage, the right tolerability. You expand it across a wide array of populations, looking at different subtypes of epilepsy to find out what works. And then you expand the usage of that compound to other larger markets. Markets where the animal models may not be so predictive, where the clinical trials are less reliable, but where the market size and the commercial characteristics make it so that once you already have a molecule, you can manufacture it, you have a cash flow, you can then expand into those markets. So why should industry keep investing in epilepsy? Because it's a place where you can win, where you can do good. You can make a difference in the world for patients and do well at the same time for your investors. Thank you.
Very, very well put. And we will hear more about industry's thoughts on investing in epilepsy as the conference continues. And now I would like to invite up the 1-2 punch from representing NINDS. First, it will be John Kane, who is the program director and works with the anticonvulsant screening program, and then the director of the anticonvulsant screening program and the winner of this year's Accelerator Award for everything that he's done in the field of epilepsy, Steve White. So we'll start with John.
Thanks, Jackie. Great talk, Chris. And you made a lot of points that I think are important for understanding our talk and why the anticonvulsant screening program has been so successful for 40 years. So Steve and I are doing a tag team approach. I'm going to give a very brief introduction and let him really spend a lot of time covering some of the exciting scientific advances and new model development that are taking place in the anticonvulsant screening program. So let's see if I... Okay. So the anticonvulsant screening program has its home in the NINDS Office of Translational Research. And the mission of the OTR is to facilitate the discovery and development of new therapeutics for neurological disorders, including epilepsy. So this slide gives you a sense of where the ASP program lies within the context of all of the programs in the Office of Translational Research. We have many programs which fund studies, research from basic research and target ID all the way to first in human. The anticonvulsant screening program has another component in which we're looking for agents as countermeasures against chemical threats, but I'm not going to talk about that part of the program any further today. So the ASP provides services and expertise to investigators who are interested in discovering new anticonvulsants. It was established back in 1975 and is the sole remaining part of what was the larger anticonvulsant drug development program at NIH. So the program provides, for free of charge, compound testing that's performed through a contract mechanism. And this mechanism is currently through the University of Utah, and Steve White is the principal investigator on that contract. The program uses a battery of seizure models in which compounds are evaluated for their potential in treating epilepsy. An important part of this program has always been that the supplier who submits compounds to the program retains the intellectual property for those compounds. Furthermore, once they enter into the program as an official participant, confidentiality is maintained. So this is important for further development of their molecules. So since 1975, there have been a total of about 580 participants in the program, and they have submitted over 31,000 compounds, and these have come from 38 countries, so it's really truly been an international effort. So roughly a 60%, 40% allocation of submissions from academia versus industry, and a little minor contribution from government as well. So this slide comes from an article by Wolfgang Loescher and describes the history of anti-seizure drugs. And you can basically see that there are 3 generations of agents. And the vertical dashed line that begins at the year 1975 indicates the start of the anticonvulsant screening program. So if you focus in on that 3rd generation of agents beyond 1975, you can see that the ASP has contributed to the advancement of a number of these compounds, as indicated in red, and those in which we had a major contribution are indicated with the large red arrows. So several years ago, the NINDS Council commissioned a working group to evaluate the anticonvulsant screening program. And that report came out in February 2012. It's available on our website if you're interested in looking at it. But in that report, they recognized the ASP for its really major contributions to the discovery and ultimate development of a number of anticonvulsant drugs. But the report also identified, as Jackie mentioned, areas of really remaining unmet medical need in epilepsy, and indicated that the program should adapt to begin trying to address these areas of unmet need. So over the past couple of years, the ASP has been involved in a number of efforts to help discover new agents that will hopefully help overcome pharmacoresistance. That is 30% of patients who don't adequately respond to current medications. We're looking for treatments that will have a better side effect profile than existing agents. And of course, we're very interested in trying to prevent or slow progression of epilepsy, for which there really are no current treatments. And we're also interested in this difficult question of treating comorbidities, such as depression and cognitive impairment or slowing. So this is an overview of the sort of the evolving ASP testing workflow. So compounds come into the program. They come in at enter in at what we call the identification phase. And we typically run the sort of workhorse acute models, maximal electroshock, 6 Hertz test, as well as a mouse model of corneal kindled seizures, and do a little bit of behavioral toxicity evaluation. And based on the outcome of these tests, if there's no activity, the testing is stopped. However, activity of compounds leads to a progression to what we call an initial differentiation phase, in which we begin to look at the compounds in other models, sometimes more resource-intensive models, but we're particularly focused on models of pharmacoresistance. And again, activity at that stage leads to a progression to a full differentiation phase where we're looking at trying to fill out the profile of the compounds and look at some of the more subtle aspects, for example, cognitive profiles of the compounds. Do the compounds have a better side effect profile, as well as looking in models of, in more resource-intensive epileptic rodent models. So in addition to this, I'll call it a standard track approach, in which we're using a serial screening protocol to make go, no-go decisions, we've also implemented what we call a special projects track. And this is really intended for drugs that might have a mechanism of action that would not be necessarily detected in our early acute screens. And an example for one example would be drugs which reduce neuroinflammation. So we've introduced this special projects track really to add some additional flexibility to our program, which we think is really critical to help us better address the working group recommendations. So I just wanted to end by talking briefly on our Panache database. This is a publicly accessible database that contains non-confidential information and data that's been generated by the anticonvulsant screening program. This is an attempt at transparency, but also an attempt at sharing data with the world and helping facilitate the discovery process. This database was publicly released at our December, or the last December AES meeting, and we initially released a batch of about 100 reference compounds, and just recently released another 50 compounds to the database. And the release will be an ongoing process over time. I don't have time to give a demonstration, but there is the URL to the link to the website, and I encourage you to get on and check it out. It's a really nice database, and will tell you a lot about the details about how the ASP runs. So I'll just end by briefly acknowledging the ASP team. This is importantly a collaboration between NINDS and the University of Utah, but also within NINDS, there are collaborations between our core ASP group and other parts of the department, as well as the extramural program directors who are in the epilepsy cluster, and Brandy Furman, Randy Stewart, and Vicki Whitmore, and Brandy's at the meeting today. And then just finally, I'd like to end by saying that I did want to take the opportunity to congratulate Steve for his receiving the Lifetime Accelerator Award. And I'll simply say that his dedication and contributions to the ASP have really been superlative, innovative, and unquestionably critical to the success of the program. So on that note, I'll hand it over to Steve.
Thank you, John and Jackie, and for the opportunity to tell you a little bit about where we're headed, if you will, and in a large part, it's in response to the working group report that John referenced just a few moments ago. If I could have the first slide. And I'm going to have just a few minutes really in the time that I have to give you a snapshot of some of the models that we're in the process of developing and characterizing and perhaps bringing online, if you will, with regard to the tracks. And I'll spend just a moment about the intrahippocampal kainate model of mesial temporal lobe epilepsy, a battery of tests to evaluate cognitive reliability. I'll spend a little bit more time on a etiologically relevant virus model of encephalitis-induced epilepsy, and a new approach to the chronic delivery of anti-epileptic drugs to epileptic rats. So this is the only slide I'm going to show with regard to the mesial temporal lobe epilepsy mouse. We put out an RFP last year about this time looking for a competitive process which we could award a subcontract through the University of Utah to bring online a different model, a chronic model of mesial temporal lobe epilepsy. That contract was awarded to Synapcell about October this time last year. And they induce epilepsy in a mouse with intrahippocampal kainate. And after about 21 days, these mice develop spontaneous epileptiform discharges. And the outcome measure there is the effect of a drug on these discharges. And it has a degree of pharmacoresistance. Some drugs work and others don't. We have received the results back from our first round of submitted compounds, and indeed we've been able to confirm through that blinded process some of the data that's shown here, in which we see activity with some of the standard drugs, but it may take high doses to get it, as opposed to some of the other drugs that have been shown to be effective in this model. We are expecting our results from a second round of testing next week, actually. So we're excited about this partnership and where it might take us with the further differentiation of compounds. As John mentioned, in the differentiation phase of the program, we are now taking some of the most promising compounds and looking for the liability for cognitive impairment using really a multi-model approach in which compound might be tested for its effects on long-term potentiation in a slice model, hippocampal slice model, its effects in vivo on Morris water maze performance, and also its effects in vivo on novel object or place recognition. And what we've learned from this so far, looking at some of the 9 of the standard anti-epileptic drugs or anti-seizure drugs on the market, is that no single test is likely to identify the full potential for cognitive liability. And so we're taking this sort of multi-prong approach, really trying to get a sense of what the overall effect might be. And perhaps maybe next year we'll be able to show you some of that data as we pursue that. So just a moment or two about the Tyler's murine encephalomyelitis model, and we've done a lot of work. My colleague Karen Wilcox and I and Bob Fujinami, who actually originally made the serendipitous observation that C57 black mice, when injected with Tyler's murine encephalitis virus, produced or started to have spontaneous handling-induced seizures at about 3 days post-inoculation. And these acute symptomatic seizures resolved by day 8 post-inoculation. They're associated with a very large rise in TNF-alpha and IL-6, 2 cytokines, so it's in response to an inflammatory storm, if you will. There's a long-term reduction in seizure threshold, and then after about 2 or 3 months of a latent period, the animals, about 50% of the animals that show these very acute seizures go on to develop epilepsy. And they also show an increased rate of kindling acquisition. And more recently, a student working with Karen Wilcox, Anthony Umpierre, has shown that there is a cognitive decline and anxiety-like phenotype associated with Tyler's virus. And these behavioral changes are certainly associated with some histopathology and changes in the synaptic physiology of the brain that's listed on this slide. So this is the mouse, and I'll show you just very briefly a seizure. This is a typical seizure that we see beginning at about 3 days. He'll stand up here in a moment. You'll see some sort of focal-like seizure activity where one paw, there's the right paw, will have some clonus, and then that will transfer over and become generalized to the left paw. And this would be a typical stage 5 racine seizure. And that is one of the outcome measures that we're looking at. So just very quickly, if you look at the top right bar there, you can see the number of animals or number of seizures per animal. It's peaking at about 4 days post-inoculation. By 8 days, this is gone. And again, the seizure severity increases over the course of those first few days, but by 8 days, all of the seizure events are gone. So the outcome measures that we're really looking at is whether a drug, be it a standard anti-seizure drug or some of the novel drugs that John referred to, is having an effect on the distribution of seizures, the severity of seizures, and hopefully, as we move forward with this, the long-term cognitive decline associated with this virus-induced seizure. So I want to show you just a snapshot, if you will, of the kind of data that we are collecting from this model with, and showing you 2 drugs, valproic acid, carbamazepine. We treated the animals twice daily with 200 milligrams per kilo of carbamazepine or valproic acid, 20 milligrams per kilo of carbamazepine, and these are doses that are pretty therapeutically equivalent to their maximal electroshock ED50. And then we assess handling-induced seizures twice a day. And the injection is on day 0. We follow them out for 6 to 8 days. And I'm not going to show you the disease severity at 15 days post-inoculation for the sake of time. But what you can see is if we look at the total seizure burden or the average seizure burden in the vehicle versus carbamazepine, it's basically the same. There around 22, and then with valproic acid has a marked effect on that seizure burden. And then similarly, if you look at the total number of stage 4 or 5 seizures during this course of evaluation, 7 days, valproic acid looks very good in relationship to carbamazepine. So we are seeing this differentiation profile as it relates to here. And then if you go on and you look at the proportion of animals who seize, about 57% of the vehicle animals had a seizure during this symptomatic phase. What you also can hopefully see, appreciate in that middle bar with carbamazepine, much more higher frequency, 85%, went on to have a seizure, and valproic acid was the same as vehicle. If you look at the odds ratio down on the bottom of this, what you can also see is that the odds of having a severe stage 5 seizure was much greater, if you will, in the carbamazepine treated group. So if they were going to have a seizure, they're going to have a severe seizure compared to the vehicle. Whereas valproic acid, they may have had a seizure at the same frequency, but the severity or the intensity of that seizure was much less relative to the vehicle. And that's shown here. When you look at the distribution of seizure types, you can see that in both vehicle and carbamazepine, the green there, about 80%, 85% in the vehicle, 80% in the carbamazepine, of those seizures were stage 5 seizures, as opposed to the valproic acid treated group. 88% of those were what we call freezing. You're handling them and they're basically they're freezing. We don't know if that's really a non-convulsive seizure at this point. We'd have to do EEG to determine that. So in Tyler's virus, as I said, it's associated with many behavioral phenotypes. It's also associated with cognitive decline and an anxiety-like phenotype. The preliminary pharmacology suggests that some of the phenotypic alterations can be modified by minocycline, an anti-inflammatory. I haven't shown you that, and valproic acid, but it has the potential as a model for assessing the prospective disease-modifying therapies of some of the novel drugs, and that's in direct response to the working group report. So in the last few minutes that I have, I want to turn to the chronic administration of an anti-seizure drug, looking at a specific model of acquired epilepsy. And anybody that has worked with rodents and doing chronic studies know that they're laborsome. You know, you can only push your graduate students so far. So one of my graduate students, shown here in the lower right, Kyle Thompson, is a bioengineering student, and I've been trying to teach pharmacology and biology to Kyle, and Kyle's been trying to teach engineering to me. So I'm not sure. I think I'll succeed in that quest. And so Kyle developed or designed a system in which it's a computer-controlled automated system in which drug-laden pellets could be dispensed to chronically epileptic or epileptic animals in a fashion that ultimately what we want to do is do this according to the PK of the drug. And he basically, we bought an automated feeder system. He tore it apart and said, I can make this better. We know that he has an infrared sensor across that little tube shown there on the far left, in which we know how many pellets are delivered, and the system will continue to do that. So the experiment is that an EEG electrode is implanted. The animals are given 1 week to recover. Then they're given the low-dose Keynote treatment according to Hilliard and Dudek many years ago, and they develop status, or an acute status. They're then 1 week, they're allowed to recover. We start monitoring them, and the diet begins to be delivered through this automated feeder. We wait until we see that first convulsive seizure, and then the animal is blindly enrolled into a study, and that study can go on for 6 to 12 weeks until it completes. The animals are monitored 24/7 with video EEG, and the previous 24 hours of video EEG is reviewed daily to identify their first convulsive seizure, and then that newly diagnosed epileptic rat is enrolled into the study. And the outcome measures that we're routinely studying is time to the nth seizure, the seizure frequency and severity, and the average seizure burden. And I want to show you one proof of concept study, an adherence study. And here we can control when the animal gets their drug and in the food. And the drug in this case is 75 milligrams per kilo delivered orally 4 times a day of carbamazepine. And so you add that up, that's 300 milligrams per kilo daily, and that's a relatively high dose compared to what is used for maximal electroshock. Shown here are the results from 2 animals in each of 3 experimental groups. The top 2 come from the non-adherent or the 0 medicated. The middle 2 come from a group in which 50% of their meals were medicated, and the third group, the bottom 2 animals shown here, is 2 animals that received carbamazepine with every meal. And I think the black dots there indicate the unmedicated meal. The red dots, red crosses indicate the medicated meal. The blue circles represent every time a seizure was observed, both behaviorally and electrographically. And what you can see is all of 6 of these animals were lined at the time of enrollment, and they were monitored for 42 days after that. So epilepsy develops after status differently in every animal. We had an animal there that took almost 20 days before we started seeing the first seizure, and that's the animal second from the bottom. It had a cluster of seizures before. The top animal, let's just take him. It only took about 5 days after status epilepticus before he started to show spontaneous seizures. You can see, based on the clusters of seizures across there, that was a pretty severe epilepsy for that animal. And you can see also that in those middle two animals, where they were getting their drug and food randomly, 50% of the time, that it's giving you very poor seizure control, much like you might predict in a non-adherent patient population. What everybody wants to be is that bottom animal, 100% seizure control over the course of 42 animals. But you can see the animal right above him, who got the same treatment, had multiple seizures over the course of that 42 days with a reasonably high dose of carbamazepine. So this is the data that's summarized from that experiment, a total of n equal 9 in the control group, 0% adherence, n equal 10, and the 50%, n equal 11 in the 100% animals. You can clearly see that 100% treatment every meal had a significant effect relative to the other two groups. You can also see from the bar graph on the right there that their seizure severity was lower in the 100% group. And if you look at the survival plot, time to the third convulsive seizure, the animals that were in the 100% treatment group stayed in the study much longer, well beyond those animals that were in the 0 or the 50% group. So to summarize then, this approach to chronic dosing can be used according to the PK, hopefully in the future. You can begin to think about a PK/PD study in a newly diagnosed epileptic rat. You can also take animals that have had chronic epilepsy for weeks to months and begin to look at the effect of a drug. So the other point that I should have made, and I can come back to this, is you can rapidly identify the responder and the non-responder. And despite the fact, if we come back to this study, the animals in the red bar, the 100% group, despite the fact that we had one animal, we only had one animal that was fully controlled. So even at this high dose of carbamazepine, these animals could be considered therapy resistant to carbamazepine. So you can go into a study looking at the effect of your new drug on these non-responding population. We're beginning to do crossover studies and add-on studies with a second drug and metabolically induced epileptic rats. You know, as it stands right now, most of the studies that we do acutely, when we do add-on studies, they're in non-induced animals, so you don't have a sense of what that interaction might be. And then long-term, you can begin to treat the animals immediately after status and treat them chronically and begin to ask the question whether your drug has any impact on disease modification in this particular model. So ultimately, the model, we hope, can help to or serve to differentiate promising investigational drugs. So what I'd like to say is, you know, I haven't had a chance to really go through these in a lot of detail, but these four models or these four approaches that we've talked about are going to aid in the differentiation of the most promising compounds that are submitted to the ASP. I think the automated feeder, thanks to Kyle's incredible amount of work in developing and writing the code that controls all of this, delivering the food, is going to be an important add-on for the stress-free delivery of drugs. And I mean, that is really important because every time you handle those animals, you're inducing stress, which can affect seizure threshold. And these models and approaches are directly addressing the recommendations of the working group report and expanding the testing armamentarium of the ASP. So again, just to acknowledge the many people involved in this project, my colleagues at the University of Utah, who, without their effort over the almost 40 years now, none of the changes that have taken place wouldn't be possible. And of course, the dedicated commitment and support through the contract mechanism by the NINDS. And with that, I'd like to thank you very much for your attention. Thank you.
Very exciting. And I just wanted to say, probably many of you have noticed, we're about 15 minutes behind time. We started a little late this morning. So what we're going to do is we'll shorten the break to 10 minutes, and then we'll take the rest of the 10 minutes from the lunch break, which is relatively long for that reason. So that will be the plan. So the last speaker of the morning session before the break is Roger Porter, who is the Chief Scientific Officer for the Epilepsy Foundation, the co-director of this meeting, and has put an enormous amount of work into it, and is going to talk to us about the accomplishments and challenges for epilepsy therapy.
Thank you very much, Jackie. It's very difficult to follow such a neat scientific discussion that Steve White just gave us. And so I'm not going to do that. I'm going to jump to the high level and talk for just 10 minutes about where we are in the science of finding new therapies for patients with epilepsy as the Epilepsy Foundation, now jointly the Epilepsy Therapy Project and the Epilepsy Foundation, now called the Epilepsy Foundation for the last 18 months, has moved forward. I need to figure out next. Let's see. Next. Oh, maybe I didn't have it up high enough. So here are the things I'm going to talk about, and it's going to be very rapid because there's a lot to talk about. We could talk about this for hours, but we obviously don't have the time. So we're going to discuss first the challenges, the ideas, the lab, the people, and then we're going to talk about each of these other things. And so you'll see this slide each time that I'm changing subject. One of the things that we really need to think about for our drug therapies is the valley of death. This is best described in the Michael J. Fox video, if you ever go on the Parkinson's website. And they describe how they fund things and they keep this valley of death from happening. I'm going to discuss this later on when I present one of the drugs later on. But they do the job right. The issue is complex and varies from product to product. Drugs and devices are different. We're very good, actually, at supporting devices near the end of their cycle because of the way this process works. We can't do that for drugs. It's $50 to $100 million in phase 3. That's completely out of our range. What we can do is address the valley of death, okay? The point from screening to an IND, okay? It's $1.5 million. That's what the valley of death is, and that's where we need to be. And our new therapy grants address the issue in principle. We just need a little bit more resources. By the way, we have, in fact, been very active, and this is just the last couple of years on the Epilepsy Foundation letters of intents received, those that were accepted by our committees, and those that were funded. We're currently in the cycle. We have some very exciting grants that have just come through, and we're very proud of this track record, which has always been a joint effort, actually, between the Epilepsy Therapy Project and the Epilepsy Foundation. In fact, if you look at the pipeline on epilepsy.com, you'll see that of the 123 projects, some of which are relatively dormant, but most of which are still active, 60 of these were supported by the Epilepsy Foundation or ETP together. So we are making an impact, okay? There's no question we're making an impact. Are we taking enough risks? Well, that's a separate question, and I'd like to discuss that in great detail, but let me just say that big-time voluntary organizations that spend $70 million a year, like Leukemia Lymphoma Society, spend at least 20 or 30% of their money on extraordinarily high-risk studies, looking for that one chance that something might really be a breakthrough.
So how do we compare to Michael J. Fox and others? Okay, and if you look at this diagram, which I've shown before, this is from 2011, but the numbers haven't changed that much. You can see that Parkinson's disease, largely Michael J. Fox, spends about $60 million a year. Muscular dystrophy, about a little over $40 million a year. Multiple sclerosis, $40 million. Epilepsy at $5 million. These are the voluntaries. So we have some work to do. Okay, we have some work to do. It doesn't mean that we can't catch up. As a matter of fact, if you look at Michael J. Fox 10 years ago, they're right where we are now. So it didn't happen overnight, but we have a lot of work to do to be really effective as a voluntary. So how well funded how do these well-funded voluntaries accelerate new therapies? I'm just going to give you one example, and again, coming back to Michael J. Fox, and they had 2 different clinical rating scales. And this was causing confusion at the FDA. It was causing confusion among investigators. What are we going to do? Because we don't know which of these clinical scales is the right one. What they did is they initiated, with their own money, a clinical trial to show which of these scales was the right one to use, okay? And they solved the problem. So they drive the clinical research. That's what we want to do right here for epilepsy, okay? That's what we want to do. That's our goal, okay? So are we set up to do the job? That might be the next question, okay? We have our new therapy grants, our Shark Tank, our SEALs, okay? Are we really set up? Yes. We have the mechanisms. We have first-class program for our new therapy grants twice a year. We have first-class program for our innovation SEALs, Epilepsy Innovation Seal of Excellence Awards for projects that we frankly would like to fund but can't, okay? We're in our exciting year of our third year of our Shark Tank, which is Jackie's invention, which is fantastic. Our first-class website with our pipeline and some really terrific people like Ken and David in the back there who have made it work. First-class meetings in San Francisco and Jackie's meeting in Florida. First-class Epilepsy Foundation leadership, okay? So we're set up to do this. Do we have the right kinds of committees? How about the SAB and the BAB, okay? Or do we have the right kind of committees to do this, okay? Yes. I won't go into the list of people that help us as volunteers on the Scientific Advisory Board and the Business Advisory Board to tell us which of these projects ought to be funded, but we have first-class people. It's absolutely amazing the energy that these people have to help us, and they're the very best. We need to take more risks, okay? If anything, these committees are too conservative, partly because we don't have as many resources as we'd like, but we need to think more about risk-taking as we get bigger and better, which of course I'm hoping is going to happen. So finally, how innovative can we be, okay? Well, we have no lack of new ideas and projects. You can see them in the program that's right in front of you. We're prepared for innovative clinical trial designs. You heard all about that from Russ Katz and all of the things that many people, especially Jackie, have done in this clinical arena. We invest in platform partnerships, as we have with Pfizer and Intellimedics. You'll be hearing some about that later in the program. And we're beginning to launch efforts in crowdfunding. Okay, we are actually not just having new ideas in science, we're having new ideas in funding. So, final slide. We have a massive underserved population of persons with epilepsy. Only 2 out of 3 patients, as Jackie pointed out, have adequate therapy, and even there with some problems. We have the teams and the mechanisms to leap forward and make big progress here. In order to make this leap, we need more research resources, okay? And so the new combined Epilepsy Foundation and Epilepsy Therapy project, now 18 months old, it's not quite new anymore, gives us new hope for the future, and I'm very proud to be a very small part of this operation as we move forward. Now, Jackie, what time are we going to resume? I suggest 10:35.
That is absolutely correct.
10:35, I'm going to start the program because we have 13 projects to present before lunch, okay? See you then.